Hi, so this is chapter 4, a tour of the cell. We're going to be looking at what a cell is, what it's made of, and the first part of this lecture is basically how we study cells and how we see them, which is using a microscope, right? So let's take a look at the first slide we have. Microscopy is using a microscope to study small objects. So small objects being cells themselves, or we can have tissues, which is a collection of cells. If you're going on to anatomy, you're going to be studying tissues in the first couple weeks of class. But you guys should know that out there in the world, there are single-celled organisms. We touched on this already. There are organisms out there that are just one cell. So bacteria, for example, are just one cell. This little guy looks like a ciliate. He's got, and we'll talk about this too, the cilia are these little hairs on this guy's body. There's all kinds of little tiny microscopic creatures that live in the water that only have one cell as their entire being, right? And then you have your multicellular organisms that are bigger than one cell. So anything larger than one cell is considered multicellular. And the really big difference besides the number of cells that these guys have is the fact that in a single-celled organism, this one cell can do everything for this organism. It uses energy, and by that it like feeds, it can reproduce, it can do all the biochemical processes that it needs to survive. On the other hand, if you look at something like our body, the multicellular organism, our cells have become specialized, right? So our bodies are made of many cells. I mean, and by many, for us, it's like trillions, right? So a lot. But our cells have specialized. So they're different from each other, and they only have one very specific job. So if you think about in this picture, the blood cell, like your red blood cell, doesn't do anything else but carry oxygen to your body parts, right? So it just travels around your body, it binds oxygen, and then gives up the oxygen. The blood cell is tied to just that one job, and it depends on the rest of the body for its survival. If you think about the heart and a cardiac muscle cell, those cells they can't digest food. They don't have a nervous system. They don't have the ability to break down the food that we eat, right, to extract the energy. So it depends on the entire organism to survive. So that's the main difference. In a single-celled organism, this one cell can do everything that needs for life. In a multi-celled organism, the cells have become very, very specialized, and so they all have different jobs for that larger organism, and they've lost the ability to sort of survive on their own. Okay, so hopefully that makes sense to you. So now let's talk about the microscope. So when you talk about microscopes, right, so what do you need to study cells? And that is a microscope. There are different kinds of microscopes out there. We're going to talk about two. We're going to talk about number 1 and number 2 in your outline. Number 1 is light microscope, and number 2 is the electron microscope. They're very different. What you're looking at in the slide here is a light microscope. So unfortunately, because we are online and we don't have lab, you will not be able to use a microscope. And that's kind of like the downside of these online classes. One of many is that you don't get the hands-on practice. However, I will give you guys a couple of videos to look at and some questions to answer about the microscope in the lab part of this class. So a light microscope. Let's talk about the first two definitions here, magnification versus resolving power. So you guys are probably very familiar with resolution because our digital age, like everything that we do is on a screen, and we know that resolution is the clarity. So the resolution is defined as the ability to see two items as separate discrete units, so that if you have two small dots on your screen, there's good resolution, they're going to show up as two separate small dots. It doesn't get blurry and then you see kind of like one big dot, right? That would be bad or poor resolution. So the crisper and clearer and the more defined the points are on a screen is the resolution. And magnification just means the size of the image, right? So you can see that these two pictures of a dog, resolution is very clear here. So this has good resolution, this has bad resolution, even though the magnification is the same. So these are things to think about when you blow things up. When you magnify a cell, there is the resolution, and it turns out that the light microscope has a limited resolving power. So you can only get so big with clear resolution. And then after that, and it's that size is 1000. So if you look at your outline, letter C, you can only magnify with a light microscope up to 1000 times. Above that, the image is blurry, or you can think of the resolution power goes down. Okay, so let's take a look at the light microscope. So these limits of the light microscope, 1 through 5, is also on your outline, so there's no need to copy this down. With a light microscope, you can only see small cross sections of a sample. So let's take a look at the picture here of your cheek cells, right? These are, if you scraped the inside of your cheeks with a toothpick and you put it on a glass slide, looked at it under a microscope, it would look like this. So your cells of your cheek are kind of round, or they kind of look like a fried egg. There's a dark dot in the middle, that's the nucleus. But you can only see one cell, and you can only see a cross section of that cell, because in order for the light microscope to see things, you actually have to, let me go back to the microscope here, light needs to shine through the object. So if you look at this flat black area, that's called the stage, and we're going to put their glass slide here. There's a hole in the stage. So the light shines from this light bulb down here. It goes through this structure, which is called the diaphragm. It goes through this, it goes through a piece of glass called the condenser. The condenser focuses that light onto your object. In this case, it would be your cheek cells. And then that needs to shine into these objects. These are called objective lenses. And then it hits a mirror and it goes into your eyes. So whatever you see has to be thin enough so that light passes right through it. And so it's really, really thin, right? And then it hits your eye. So letter A, you could only see small, thin cross sections of samples. That's a limit. You can't see the whole, a chunk of tissue that's too thick. It's just not going to work. Letter B, you can see the cell and large organelles like the nucleus and chloroplasts, but nothing smaller, because I said, remember, you can only magnify 1000 times. So we can see that dark dot in the middle here, right? And when you add a stain, you can even see it better. That's the nucleus. Letter C, you can only magnify up to 1000 times. Letter D, the resolving power is limited. You can't resolve anything smaller than 0.2 micrometers. And micrometers, remember that micro is 10 to the negative 6th of a meter. So let's take a look at that picture. So this is an example of your field of view. When you look through a microscope, you see an image that's in a circle. And then you're going to look at the objects here. So on this side, this says resolvable, meaning that the objects are bigger than 0.2 micrometers, or they're spaced out farther away than 0.2 micrometers. So here is every little tiny hash mark is 0.2 micrometers. So if the objects are farther apart than that, or they're bigger than that, then these are resolvable. However, if you have tiny objects and the two fall inside of 0.2 micrometers, meaning that they're closer together than that, or they're smaller than 0.2 micrometers, then what happens is this little two circles will look like one big blurry circle. These two objects here will look like one big fat oval. So it's very difficult and not resolvable to see these smaller items. So that's a limit. You can't use the light microscope then. So if you knew that you wanted to look at something smaller, like a virus, viruses are super small, then you can't use a light microscope. You need to use another kind of microscope that's fancier. Okay. Letter E, if you look at the need to stain, this is this picture here. So if you were to put your cheek cells on a glass slide and look at them, they would appear basically see-through, right? Because your body is basically not colored except for your hair and like the pigmented parts of your body, but your cheek cells are going to be clear. And when you take them out, you look at this. So this is not very helpful. Sometimes it's very difficult to see. So you always want to add a stain. And a stain is a colored substance, kind of like food coloring, but it's fancier, like methylene blue or iodine. But those will put a color onto your cells so you can really see the outline of them. You can really see the nucleus. Much easier to see. All right. The nice thing about the light microscope also is that you can actually see really small living things. Remember this guy here that was swimming around? You can see that with the light microscope, and you can see him swimming around. So if you just had like a little glass slide with some water on it, a tiny drop of water, You can't see this guy with your naked eye, but you can see him with a light microscope. So that's good for the light microscope. Okay, and I also see the lab manual for details. So since we don't have a lab manual, we're going to just be using the Canvas lab activities for that. Okay, so that's the light microscope. And then this is just a scale for what you can see. So what's visible with your eyes, right? These are the measurements. So 10 meters, 1 meter, 10 centimeters, 1 centimeter, frog eggs fall something around a little bit bigger than 1 millimeter. And then there are objects that you can see with the microscope. You can see plant and animal cells, you can see the nuclei, you can see most bacteria and mitochondria. So you can see those items with a microscope. But anything smaller, so in here, atoms, small molecules, lipids, proteins, ribosomes, those are too small to resolve with a light microscope. But there's pictures out there. There's pictures of viruses, there's pictures of lipids, there's pictures of small molecules. So where do we get those? So that is number 2, the electron microscope. So before we move on to electron microscope, what is a micrometer? So this is going to go back to your lab activity 1. Do you guys remember what is a micrometer? It's answer letter D as in dog, 10 to the negative 6th of a meter. All right, so this is an electron microscope. So take a look at this very different thing. It is an object that is much larger than your light microscope. You have to sit at this desk. There's a long tube where electrons are beamed down onto your sample, and there's a computer to pick up the image. Because our eyes have evolved to use light to give information to our brain, our eyes don't pick up electrons. So we can't use our eyes and look in here. We have to rely on a computer screen. So electron microscopes can see very small objects, excuse me, like viruses and molecules, much smaller than a light microscope. So these items are much smaller than what a light microscope can see. That's what I meant by that sentence. It uses a beam of electrons that bounce off the object, and the diffraction pattern is detected by a computer that generates the image. Okay, so let's look at some of these. There are 2 kinds of electron microscopes. All right, there is one called the transmission electron microscope and the scanning electron microscope. So I know in your notes these are kind of switched, but let's look at the first one, transmission electron microscope, TEM. This picture here and this picture here, those are all TEM pictures. They are 2-dimensional. They are a cross-section, right? So that you can actually see through the object. And they give you highly detailed images of things inside of the cell. So this is a picture of 1, 2, 3, 4, 5 cells kind of together. And I can see inside these cells, there's little units, these little pockets of things. On top of the cell, there are these little hairs. Okay, so actually this picture here, this is a picture of cilia, which is a hair-like structure on the top of cells. This is the same thing. This is cilia, but cut in a cross-section, and I can see the inside. Okay, so this is a cool picture of a virus that only infects bacteria. It's called a bacteriophage. And I can see inside, I can see the structure here in the tail fibers. So the transmission electron microscope is very good for 2-dimensional internal structures, right? Those images. But if I look at my scanning electron microscope images, this and this, what kind of image does a scanning electron microscope give me? It gives me a 3-dimensional image. I don't see the inside, but I can get very good detail on the external surface, right? This bacteriophage, these two are the same thing. I can really see the 3-dimensional object here. Really spooky looking thing, right? This bacteriophage. Now for the rest of a few more slides, I just have some really fun, this is just for fun, pictures taken with a scanning electron microscope, the 3-dimensional images. So this is a fly. This is the eyes of a fly, and you can see the great detail of the different units of the eyes and the hairs. This is the antenna of the fly in the socket going into his head. So really, really great detail here. This is pollen. So when you take a breath of air and you're outside, you're breathing in all kinds of material into your lungs. You're breathing in dust, you're breathing in mold spores, you're breathing in, we know that we're breathing in other people's respiratory droplets, that we're talking to. And they're also breathing in pollen. So this is made by plants. This is what the plants use to fertilize themselves, right? And you can see how different they are. So this is why you can be allergic to one kind of plant and not another, because your immune system can really see the difference of these different pollen grains. So you might be allergic to this really big guy here, but these little small ones, these don't bother you. And you can see that they're drastically different. Okay. This is a lice nit that's attached to a hair. This is the egg, right? This is the nit. And then this is an interesting part. This is called the operculum, and this is where the baby lice will, or I say baby louse, will pop open when it hatches. This opens up like a submarine, and then you can see the baby louse will climb out. This is DNA. This is bacterial DNA. Bacteria has a DNA that's a circle. And then human DNA is like this. So this is a very compacted form of human DNA. We'll talk about this a lot later, but this is a different chromosomes. So you can see that the DNA in bacteria and the ones that we have are very different. I should fix that statement by saying the overall shape that they take is different, but the actual molecules that make the DNA is the same. Okay. This is skin. So this is human skin. And notice that we have this pink layer and a green layer. The pink layer is the epidermis. The green layer is the dermis. But you can see how it looks just like it's sloughing off. Right? These cells are flat, they're dry, and they come off your body all the time. And the green part here is the dermis, the deeper part, and this is where you have all the collagen. Here's a pore of your skin, taking with an SEM. Here is a mosquito head, and you can see the sharp proboscis coming out here. So I believe it's this. I think this is the sharp end of it that comes into your skin, and the blood comes into the mosquito through this structure. All right, so that was the fun pictures taken with the scanning electron microscope. Now let's start with the parts of a eukaryotic cell. So I'm going to go ahead and pause the lecture, and we'll start with the eukaryotic cell in part 2.